Hello, welcome everyone. I would like to start by thanking everyone that participated in yesterday's session and extend a warm welcome to those who are starting with us today in our journey to world of bioinformatics. Paul has a doctoral thesis from University of Mississippi, and he has a Ph.D. in pharmacology and toxicology and a postdoctoral fellowship at NIH. And he was actually a certificate student. Paul did a certificate with us in bioinformatics, and that's how we know him. He's extremely talented, very knowledgeable person, has a lot of experience with immunoassays, and he's going to teach the next-gen sequencing. I want to thank you all once again, and I'll hand it over to Paul. Thanks, Paul.
Okay, thank you. This is the second course in the series on next-generation sequence analysis tools. I'll do a brief review of what we talked about yesterday so that we can bring the people who didn't attend yesterday up to speed. We're talking about nucleic acid sequencing. It's more efficient than protein sequencing because it's a sequencing by synthesis method. And the way it works is that you have a template strand and you add in just a soupçon of a fluorescently tagged nucleotide, which also causes a chain termination. And when you have this collection of fragments with different lengths with the last one tag, you can then line them up and predict what the original sequence was. So originally, sequencing was done massively parallel by having lots of technicians and lots of instruments. And the first human genome, which came out in 2003, cost over $100 million. So it was an enormous expense. And it got better over time, but there was a sudden technological advance in 2007, 2008, which caused the cost of sequencing to drop precipitously. And now, in 2020, it costs less than $1,000 to get a full genome sequencing because of next generation sequencing. So this is the dominant technology in the field now. It's from a company named Illumina. And the Illumina sequencer is an elegant system where fragments of DNA, about 200 to 300 bases in length, have a small fragment put on both ends, and they're amplified and then attached to a surface. And then this surface, then the DNA flexes and both ends attach. And after both ends are attached, the two strands of the DNA are separated, and you have a more amplification done, so you get a large spot of homogeneous DNA in a particular point. And then you do your Sanger type sequencing with the fluorescent tag. And if you look at the colors in the 4-channel below, you see green, violet, red, green, yellow, red, which translates to a sequence of TGC, TAC. So you have that's one spot, and there are lots of other spots around it which are independent sequences. Multiply this by millions, and you can do an awful lot of sequencing in one pass. So here's an image from the sequencer. And in the current instruments, you can have between 40 and 50 million paired reads at a time. And over the course of less than 3 days, you can get an output of between 13 and 15 gigabytes worth of sequence data. And this is the equivalent of reading the entire human genome twice over. So here's what the output looks like. It's basically a set of randomly distributed sequences, long stretches of A, T's, G's, and C's, and some quality control information to tell the next tell the processing systems how good the confidence on a particular read is. So it's basically a big puzzle, which you have to put the pieces back together again. And the way to do that is that if you don't have a template, you find out where the pieces overlap and make longer areas of contiguous sequence. And so you find as many overlaps as you can, and you get a contiguous sequence. And then you try and work with those contiguous sequences to get larger and larger overlaps, till hopefully you can get the entire sequence, whether it's a genome or an RNA or whatever it is. And it's made easier if you have a reference genome to work off of. It's like being able to look at the picture of the puzzle before you start. Makes it a lot simpler. And that's called alignment. So we now have a computational problem. You're working with very large files and you're doing a lot of manipulations. The files can be enormous. For example, for doing a gene-wide alignment of sequence, or GWAS, the files can be 25 gigabytes or more. For an RNA sequence for differential gene expression, the files are between 3 and 15 gigabytes. And for chromatin immunoprecipitation, they're about a gigabyte or less. So if you were to try to do this on the average 16 gigabyte laptop computer, it would run very slowly or not at all. And you would also need a pretty large hard disk. You know, a good size would be around a terabyte. In contrast to the alignment process, the analytical computations are not nearly as demanding. For the GWAS, the files that you're working with are less than a gigabyte, and the other types of experiments are proportionally lower. So it's not RAM intensive, and it can easily run on a 16 gigabyte RAM machine, and the programs are not operating system dependent and use readily accessible program languages such as R and Python and Java. So a quick solution for those of you who are trying to start out in the field is to use cloud computing for the initial steps in a server like Galaxy, which gives you access to completely free access. It has most of the programs that you'll need for doing sequence analysis and enough memory. And it's also menu driven, so you don't have to really worry about all the fine details of doing the alignment. There are also some downsides in that because it's menu driven, that you have to do things the way that they want, and it's not terribly flexible. So the strategy, but you can do your analysis on your own machine. So the strategy is to do the alignment in the cloud and then do the analysis on your own machine. So here is the website for Galaxy. It's usegalaxy.org. And as you can see off to the right, there are a whole series of different menu items for all sorts of things that you'd want to do for sequence analysis. And here's a general workflow for doing the sequence analysis. And you start out with the raw reads. You filter out the poor quality reads, put it through an alignment program, and then you put the alignment through a series of programs, either to count the RNA sequences that were in your sample, to look for places where the sequence doesn't match up so you can find where there are mutations, or to do a chromatin precipitation analysis where you get information which is in peaks. So the output of these files are then put through a statistical analysis program, such as the one that some of the ones that will be talked about tomorrow. And you get an analytical output, which you can either report or put through second-level analysis systems such as BioPython, or things that you can find in R. So yesterday we talked about an example of gene variant analysis in pharmacogenomics, where we looked at the consequences of gene variants on the metabolism of a drug called warfarin, which is metabolized and inactivated by an enzyme called cytochrome P450 CYP2C9. And there are other drugs. It's also metabolized by an enzyme called CYP2C19. And we looked at an example where the individual had a defect in CYP2C9, which decreased the activity of the enzyme. And she also had another variant which prevented the activation of clopidogrel, which is the alternative drug. So this is a person that would have to be very careful about her use of anticoagulant drugs. So now on to something a little different. Today we're going to talk about some experimental things, which are of use for both understanding what's the cutting edge of molecular biology and also for pharmaceutical development. And it's called epigenetics. DNA sequence is very much like memory in a computer. It's got a lot of information storage, but you need to have an input and an output. So you need to have control systems. And there are a number of patterns that seem to have developed that look like they're control systems. For example, if you look at the diagram here, there's some marks over here, and those are DNA methylations. And it's been found that these methyl groups that are right on the DNA itself are not randomly distributed. They're more concentrated right around the transcription start of genes. And they have a tendency to be clustered on genes that are turned off long-term in cells. In addition, DNA is rarely ever naked just by itself. It's usually wrapped around a core of proteins called histones, which are in a cluster. And these histones are used to compact the DNA and give it control sequences. And if you look over here, you'll notice that there's a little mark up here too. And on the N-terminal tails of histones, there are a bunch of positively charged groups which act as places to put little flags, such as acetyl groups and methyl groups and very small peptides. In addition, there are some interesting things that are being looked at for control, which are very, very small RNAs called microRNAs. And these are only like a few dozen residues long. And they seem to be involved in control systems like silencing and degrading messenger RNAs. And lastly, there are some non-coding RNAs. You don't seem to have any protein sequence in them. Some of them are quite long, called long non-coding RNAs. And even more interestingly, some of these non-coding RNAs are on the opposite strand to the coding genes and interact with the coding genes. So this looks like this is a place where there's room for control systems. So we mentioned that the histones get methylated or acetylated. And it's been found that when the histones are methylated, the DNA is more tightly wrapped around them. And the genes under these conditions have a tendency to be silenced. Whereas the genes which are acetylated have a tendency to be less tightly wound, and the genes in these areas are more likely to be active. So this gives us the beginnings of a control system. And cells put an awful lot of effort into making enzymes that control these markings. For example, there are a bunch of enzymes that are involved in either methylating or demethylating DNA. There are enzymes that acetylate DNA, acetyltransferases, and histone deacetylases as well. And there are also enzymes that are involved in transferring methyls on and demethylating the DNA, and also phosphorylating it as well. So on the right-hand side, we see that there are a whole series of markings that are decorating the tails of the histones. And we're going to look at how differential gene expression is regulated by these markings. And we're also going to look at how drugs can interact with this. Early on, it was found that methylation is regulated by small molecules like azacitidine, which is basically an analog of the nucleotide cytidine, and causes DNA demethylation. So that would activate genes. And there are also compounds that are histone deacetylase inhibitors. It's interesting that some of these are old compounds that are being used for something new. Valproic acid is an anticonvulsant, which seems to also have histone deacetylase activity. Not terribly specific, but it's there. And there are also a number of histone methyltransferase inhibitors. And today we're going to look at one from GlaxoSmithKline, which is called GSK 2879552. This is a histone methyltransferase 1 inhibitor. So the model that this is particularly interesting for is enhancement of cancer treatment. And in particular with a cancer called acute myelogenous leukemia. It's been found that all-trans-retinoic acid, the same molecule that's involved in helping you see, if you expose the monocyte-like leukemia cells, that they go from a round mononuclear cell to a much more complex shaped cell called a macrophage. And normal macrophages are basically the garbage collectors of the body, which one of their prime functions is to spill their guts and destroy invaders. So this whole process causes the cancer cell to die. And interestingly, compounds like the GSK compound don't really cause the cells to differentiate like this. but they markedly enhance the effect of the all-trans retinoic acid. And cells which are relatively refractive to the retinoic acid become responsive. So let's look at a model that might be used in a pharmaceutical company who's trying to find a new drug for treating some sort of condition, say like some sort of a cancer. You have your control cancer cells, you treat them with drug 1, and you may get some response. You treat them with drug 2, it doesn't seem to do much, but when you put drug 1 and drug 2 together, instead of getting 1 + 1 = 2, you get 1 + 1 = 60. You get a potentiation of the effect. And we're going to look at a model where we split the sample in two, and half of the sample goes through an extraction of RNA, reverse transcription into DNA, and then a sequencing, alignment, and statistical programs to get differential gene expression. And the other half of the sample gets exposed to a very intense sound, which caused the DNA to be sheared down to small fragments which are wrapped around the histone nucleosomes, and then captured by an antibody against the tags, like the acetylation. And believe me, the antibodies to these very small antigens are hard to make. I've tried to do it. And if you can get one, they're very profitable. They're a good source of revenue. So once you have the antibody attached to the histone tag, you pull it down by immunoprecipitation, and then you wash away everything that's irrelevant and run it through the DNA sequencing, and you get a series of piles of DNA fragments, which can be then aligned up on the genome, and it gives you an indication as to where something happened in terms of gene expression. For the experiment today, we're going to download a data set that was from a published manuscript, which was uploaded to the National Center for Biotechnology Information. This is a way of sharing data so that other people can look at the data and mine it for things that they're interested in. And we're going to use the Galaxy to do our alignment, and then take some our algorithms from the public domain site called Bioconductor, and then do our secondary analysis. So here's the data set that's in NCBI. It's a series of files where they treated the cells with the retinoic acid, the GSK compound, or a combination of both. And if you want to look at the original paper from the contributors, it's down at the bottom of the file. So here is the workflow. You take the RNA, you take your raw reads, you run it through an RNA specific alignment program. You get your aligned reads. You then put it through a counting program, which counts the number of times that any particular aligned RNA was found. And then you get a table where it has the name of the gene that it was on and the number of times that that particular sequence was found. And the level of information compaction is enormous. You're going from around 12 gigabytes of raw data down to 2 megabytes. So it's data reduction by a factor of about 3,000. And then we're going to go put it through an analysis program in Galaxy and a comparable program on my local computer. So here is the initial step in the process, the alignment, where you tell the program what genome you align it to, and you tell it the files that you're going to use for doing the alignment. And under certain circumstances, there are some advanced options that might be useful, but we're not going to use them today. And then you wait and you wait and you wait to do the 8 samples that are in this experiment and run it through both the alignment and the counting. Took me the better part of 3 days. At the end of it, you have your files in tabular form. Now, if you look at this, this image, you can see that you have your tabular files and they're in 4 different treatment groups. But if you look at it, it's actually a 2 by 2 table where you have either the treatment of retinoic acid plus or minus, or the GSK plus or minus, or the combination thereof. So in reality, when you're entering this into the program model for doing the analysis, you need to enter each sample in twice. So here we go. We enter, this is in the program called DE-seq2, where I went through and entered in the sample here into that particular place in the model twice, once for each of the different places where I treated the sample was treated with a control. Either the DMSO control for the retinoic acid or the vehicle for the GSK compound. And about 3 minutes later, you get an output. So that's really pretty incredible reduction in time. And this graph shows you that there is what the number of normalized counts looks like relative to their variability. And as you see, as the number of counts gets lower, the variability goes up, but it's a continuous function. So that's about what you should expect from this type of data. And there are 4 other graphs, the most important of which is called a principal analysis, component analysis, or PCA. And basically what this is looking at the variability of all the samples relative to each other, and you generate a cloud of variability of relationships between different genes. And if you look at the cloud, it's sort of cigar-shaped. So if you look at the distance from one end of the cloud to the other, that's one axis. And if you look at it along the narrow axis of the cigar, you have a second variable. And if we use these 2 variables as an index of that particular sample, then it gives sort of a fingerprint. And if you're looking at this graph, you can see that here's the DMSO and vehicle, and that in purple. And that's a nice little cluster. The GSK compound and the down here in green, retinoic acid in blue, and the combination in pink. And there's 4 nicely separated groups. So this is a nice experiment to analyze. And you can get at a table with significance on it, but that's about as far as you can get with this program in Galaxy, because it doesn't really have the way of doing it. There are another couple of programs that are a little more sophisticated, which allow you to do the post hoc tests, but they're kind of a little bit hard to handle. So I preferred to do it in my own local computer using a little bit of our programming. You know, it's not hard to do, so I'm going to do this. And here is our output table from the reads. And if you notice here, there are a couple of samples where there's zeros. So those are genes where nothing was present. And those are going to have to be pruned out. So now here we're into a local program on my computer called RStudio. This is a tool which can be used to run our programs. And there are a bunch of the algorithms that I've downloaded from the bioconductor site. Some of them are involved in doing the sequence analysis, some of them are for annotation, and some of them are for doing graphics. And here is one of the functions that I'm using. And this is for doing the contrast, which are doing the post hoc tests. And in these bioconductor packages, they have some very sophisticated things that you can do with just a simple call. You tell it, for example, to do the contrast fit. You tell it the source of the thing you're going to measure and what you want to look for, and then you run it and it'll give you an output. And then you tell it what you want to have as parameters for how rigid the analysis is, and it'll give you an output. And since we're going to do this a couple of times, you put it in a function. And you can call the function as many times as you want, as long as you give it the right inputs. So let's pull in our functions. Now we're going to load in some data. As you can see here, we started out with, down in the lower left-hand corner, we started out with 60,000 different gene records. And after filtering out all the zeros and the low expressed things, we went down to 13,000. So that pruned out a lot of stuff. So now let's do some modeling. We're going to do some modeling and also fit it to a statistical program called linear modeling fit, LMFit, which is a linear modeling program which you'll learn more about tomorrow. So it's generated the groups that we're going to look at. It's, again, the 4 basic treatments that we looked at, and the ways to put them into the analysis matrix. And we also have basically the same PCA plot. It's a bit squished on screen here, but it's basically the same thing. So we've run the model, and now we're going to do the post hoc analysis for getting at the specific treatment groups. And we're going to get some output. Okay, so that entire analysis from beginning to end took under a minute. And at the end of it here, I have given two tables. The first table has a number of the genes that were, how many were up-regulated and how many were down-regulated. And in the lower table, we have a series of genes. We have a series of genes that are a sampling of different types of regulation. And just looking at, for example, the retinoic acid versus control, there were 492 genes that were had a change in their levels. 374 had an increase, and 145 were decreased. In contrast with the combination treatment, there were over 2,000 that were increased, that were changed, and about half of them were increased and half were decreased. So let's look at a few genes. The first one that we'll look at is CD14. And CD14 is a marker that's found on mature macrophages. So if CD14 is present, you have a cell that's basically a macrophage. And the retinoic acid by itself didn't cause it to be present. The GSK compound didn't as well, but in the combination, it's there. Now, the next compound, the next gene we'll look at is called DHR3. And DHR3 is an enzyme that converts retinol to retinoic acid. So it's an amplifier for the retinoic acid signal. And when retinoic acid is present, you'd want to have this increased. And that's exactly what one sees, that there's an increase and an upregulation in this gene. And any place where the retinoic acid is, you see it. KDM1 is the lysine demethylase, and it's increased in the combination treatment. But a related gene, KDM4A, which has a slightly different methylation specificity, is decreased. So here's the beginning of something interesting. The enzymes that are involved in regulating these genes have some differential regulation. And here are a number of other genes, including one called P21, which is involved in looking at, which is a sensor for damaged DNA. So if you're going to be using a DNA damaging agent to kill your cells, that might be a good place to look. Okay, so back to our original model of the histone methylation and acetylation. We stated that the histone acetylation is a condition which will cause the DNA to be open enough to allow gene expression. So let's look at one of these markers. It's a methylation of lysine 9 on histone 3. And run it through our immunoprecipitation system and get a series of peaks. It runs through an alignment, a peak calling, and comparison of peaks. And when you're running this in Galaxy, you need to be careful not to just take all the defaults, because the width of the peak may vary. And here's an example where it's sort of like where you're reading from both strands of the DNA. It's sort of like having your hands face down on the table. They are inverted to each other. And the top of the peak gives you an index of where the width of the peak is. And if you look at the right, it's a single peak, which is a uniform thing. So that's nice and clean. So in the integrated gene viewer, IGV, on my local computer, I looked at a couple of the genes, and this is the DHR3. So the transcription start site, or TSS, is on the right-hand side here. So that means that the gene is transcribed to the right, and right at transcription start, there is a big pile of acetylation. It's increased in the treatment. There's not really any decrease, and the number of places where it's unchanged relative control is not really great. In the combination treatment, there's similarly an increase in the acetylation as well, and the two sort of match up. And interestingly, there's some acetylation within the gene. So it's not just the effect at the promoter that's causing this to be of interest. There's also some unwinding of the DNA within the gene itself, which is helping this gene to be transcribed. So here's the KDM4. Now, this is a gene that by the RNA expression was strongly repressed. But here we see something kind of odd. The transcription start site for this gene is on the left-hand side. So the gene is being read to the right, from left to right. And there's acetylation right at transcription start, both in the retinoic acid by itself and the combination trio. What the heck is going on? This should be something that would be turning the gene on. But what we see is a turning off. So something more complicated is going on here. And there's a subtlety. There's also acetylation at the other end of the gene. And if you look carefully at it, you'll see that it's in a region where there's a label KDM4A AS1. AS1 stands for antisense. And this is a 7 kilodalton fragment of RNA that gets synthesized on the opposite strand to the one that makes the protein. So this is on the antisense strand, and it's synthesized towards the sense strand transcript. So that's interesting. This is something that might be involved in regulation. And what's been found is that antisense do some interesting things. For example, if the two overlap, which is slightly the case with this gene, that the two polymerases collide with each other and stop. And you never get a complete RNA. You never get a complete coding RNA. So the gene is functionally repressed. Or the other possibility is that the antisense is like a person in a room who's talking very, very loud. They grab all the attention, and the sense strand doesn't get any. So it becomes a matter of who's shouting the loudest. And when there's somebody very loud, the sense strand never gets a chance to be heard. So that makes sense. You know, if the antisense is very loud in this case, then the sense strand will never get heard. And so the gene is functionally repressed. So this gives us a new opportunity for finding a way of throwing a monkey wrench into the works. We now have tools for finding out how we can find new ways of manipulating cells to make them do what we want. To summarize, next generation sequencing is massively parallel, and we have ways of both dealing with the computational challenges of sequence alignment and being able to analyze them. And we can do it with DNA and RNA samples to answer pharmacologically relevant questions for both clinical use and for research. And now I'd like to give you an invitation. I've given you some tools that you might be interested in. Come and get on the boat and learn some more about bioinformatics.